Hello everyone, let's look at control and coordination in mammals or in animals. First and foremost, what really is coordination? What do we understand by the term coordination? What do you understand by coordination? In a simple term, when we talk of coordination, coordination refers to the different parts of the body working together. So when you see the leg, the hand, the eyes, all of them are working together, that is the meaning of coordination. Different parts of the body coming together and working together to achieve something. All of those comes from movement of the impulse, that is involves the transmission of the signals. So we're going to learn of how these signals are going to be transmitted through the human body and an organ, which we're also going to learn from it. First and foremost, we need to understand the first term, that is the endocrine system. In the endocrine system, we have two things that we need to understand. One, that is the endocrine gland. Two, that is the target organ. When we talk of an endocrine gland, this endocrine gland is an organ that releases the hormones. It is an organ that releases the hormone. For example, we have the pancreas. The pancreas releases the insulin or glucagon in due situations that we might learn more. That is the topic of homeostasis. And then two, let's look at the target organs. When we talk of the target organs, these are the organs that respond to the hormones that are released by the endocrine glands. For example, the pancreas releases the insulin or the glucagon. These are going to be responded by the organs. First of them are the liver cells. Perhaps the other can be the skeletal muscles. Hope you're good with that. Then two, what we should learn from this, also from the endocrine system, is that stimulus is going to be present and the receptors, for example, the adrenal gland will release the adrenaline. Are we together? The hormone adrenaline. So this adrenaline will be responded by adrenaline binds to the cardiac cells. So that is the small example that I can teach you from that. Then secondly, I want us to understand what really is the nervous system. Most of us have heard of what nervous system is all about. Now, today, we are going to learn of what nervous system is all about. When we talk of nervous system, this is where nerves come together. In a short term, nerves come together in order to interconnect and share connection in order for a response to be brought about. As you see, the stimulus will be present, will be stimulated. The receptor cells will receive the signals and they'll be transmitted. The first neuron, the second neuron, the third neuron, then they are going to be brought back to the effectors for a response. Subhanallah. So let's look at the difference between an endocrine and nervous. What is the difference between these two? Most of us confuse when we talk of endocrine and we talk of the nervous, as I was defining for you before. Now, it's differentiated with the signals, speed, and the ATPs that are produced. When we look at the signals, endocrine systems, they produce hormones, as I described there. They produce hormones, whereas the nervous system, they produce electrical impulse. Then when we talk of the difference in terms of speed, the endocrine system, they are very slow, whereas the nervous system, they're very fast. The ATP that is produced by the endocrine, they are very low, whereas that of the nervous system, they are very high. We're going to explain a reason as to why they produce low and high, and why this endocrine produces hormones, whereas the nervous system produces the electrical impulse. So let's proceed with coordination, dear brothers and sisters. First and foremost, allow me start with the nervous system. When we talk of the nervous system, they are of two types. One, we have the CNS. Two, we have the PNS. When we talk of the CNS, that is the central nervous system. The central nervous system, just the simple structure that you should understand is this. It entails two things. One, that is the brain. Two, that is the spinal cord. Now, from that, we're going to generate the PNS. When you talk of the PNS, those are the peripheral nervous system, in which also it entails two things which are connected to the CNS. As we saw the diagram of the CNS, let's see the diagram of the PNS. In the diagram of PNS, we have seen projections coming out from the brain, and we have seen projections coming out from the spinal cords. Those are also having the special name. The projections coming out from the brain, those are called the cranial nerves. They are connected to the brain. Then another one that are connected to the spinal cord, those are called the spinal nerves. Now, what really is the function of this? The function is in order to transmit nerve impulse. Are we together? That is the main function. It's order to transmit nerve impulse. Now, let's talk of the types of the neurons. When we talk of the types of neurons, they are roughly three: the sensory neurons, the relay neuron, the motor neuron. As a common language, you and I know very well. The sensory neuron, they are to sense, whereas the relay neuron, they are to connect or to relay, whereas the motor neuron, they are just for mobility or for movement of the body. As we are well aware of what happens, what do you think? What can someone give me in a comment section? What do you think happens when light strikes an object and the eye lands on that object? What do you think happens to the CNS, the central nervous system? What happens to the relay neuron, the sensory neuron, the relay neuron, the motor neuron? What really happens when stimulus is stimulated by the human eye to see and the photoreceptors detect it? What do you think happens really? Can someone explain for me that into a simple term? Then let's compare between the sensory neuron and the motor neuron. As we have seen the diagram of the sensory neuron and the diagram of the motor neuron, I'm sure with just vision you can differentiate and you can give the similarities between the two. First and foremost, when we talk of the sensory neuron, the cell body is not within the CNS, that is the central nervous system. The cell body is not in the CNS, it is not located here. Whereas the cell body in the motor neuron, they are located in the cell body, just right here. And when we talk of the similarities, both have cell bodies, of course, as you can see here. Another one, both have axon to transmit impulse. As we see, this is what's called the axon. They're usually long. So those are the axons, as we see. Another one, both have dendrites to increase surface area. So when we look at the surface area, when we look at the dendrites, that is this one, the one that are able to interconnect to one neuron to another for the impulse to be transmitted into a longer distance. Another last one, both have terminal branches and synaptic knobs. We're going to look at more of what this synaptic knob is all about and what it entails. And that is it. In a simple language, when we talk of the axons, there is something that there are two types of axon that we should know. One, the axon that is myelinated, and then the axon that is not myelinated, that is unmyelinated axon and the myelinated axon. So we're going to look at what really those means. Now, let us look at the action potential. When we look at the action potential, we all can relate that this action potential, they are the rapid change in the electrical charge distribution across a cell surface membrane. We see that at the axon, negative charges are inside, whereas positive charges are inside. And at time t, we're going to see a change between the negative charge and the positive charge, that is between the membrane of the axon. So we're going to look at how this thing really happens. But in simple terms, we need to understand that the action potential, it is a rapid change in the electrical charge distribution across a cell surface membrane. Are we together? So as we see here, this is what will happen at the axon. We're going to see negatives inside, whereas the positives outside, and we're going to see a situation whereby positives are going to come in and negative will come out, and I will show for you how that will happen. Now, in a short term, let's learn the axon structure, how these axon structures are looking like. From among the contents of the axon structure that you should know, one of it is the sodium potassium pump, which is here. Then we have got the sodium channel and the potassium channel. That is one thing you should know. And all of this one here, they are voltage gated channel. Why? Because the channel proteins, they only open and close at specific voltage. They will ask you, why is it called a voltage gated channel? So this is also a good hint that you can think about. And then let us look at the resting membrane potential. It is usually denoted as negative 70 MV. Now, how does the axon create and maintain the resting membrane potential? So in a normal term, this diagram explains everything. First and foremost, when the axon is stimulated, the sodium potassium pump is activated. This one here is going to be activated, and the ATP powers sorry. So first and foremost, as I was explaining, when the axon is stimulated, the sodium and potassium channel are going to be active. Then the ATP is going to power the sodium potassium pump. As you see here, is going to power, the ATP will power the sodium potassium pump. It actively pumps outside 3 sodium and pumps in 2 potassium. As you see here, 3 sodiums are going to be moving out, whereas 2 potassium are going to be coming inside the axon. Voltage inside becomes lower than outside, hence resting membrane potential created. As you see, there are only 2 potassiums coming in, whereas more of the potassium, the sodium ions, will be moving out, thereby creating more positives outside than what is inside here. And what will happen is that negatively charged proteins inside the axon also makes the inside have a lower charge. The proteins that will be located inside the axons will make inside here become more negative. So that is why we say that the gradient, the sodium, when you look at the sodium outside is going to be great, where the sodium's inside will be less. But when you look at the potassium's inside is going to be more, and the potassium outside will be less. So that is a little understanding that we can gain from there. Now, let's look at what depolarization really means. When we talk of depolarization is usually denoted by positive 30 MV. Now, what happens in depolarization is that during depolarization, voltage-gated sodium channels are opened. As you see here, the sodium channels are going to be open. Sodium rushes into the axon down the concentration gradient. It rushes inside the axon. And then from negative 70, which was at the resting, from negative 70 MV, it becomes positive 30 MV. The sodium ions rushes, hence achieving the membrane potential, which is positive 30. They rush in and achieving that 30. After achieving the 30 and the positive 30, when achieving it, the channel will close. It will automatically close. In a short term, that is what is meant by depolarization. It only happens, in summary, it happens due to the opening it happens after the resting potential achieving negative 70. Then after achieving negative 70, we see the sodium channels are going to be open, allowing the sodium to rush in at time t, at a point whereby it becomes negative, it becomes positive 30 MV. So after reaching that point, we see the sodium channels will be closed. That is what is called depolarization, and that's what it talks about. Now, let's talk about repolarization. What really is repolarization? When we talk of repolarization, once the membrane, now let me explain it in this small summary here. Now we talked about this one and we said that the ATP, I'm supposed to draw here my ATP. My ATP is my power source, the one that pumps, the one that will activate my sodium potassium ion pump. Are we together? It's the one after the axon being stimulated. My ATP is going to be actively pumping the sodium and potassium pump, therefore allowing 3 sodium ions outside and allowing 2 potassium ions inside. I've drawn it here, sorry. So after allowing it inside, all of us can testify that at the resting potential, the sodium ions are more outside, whereas the sodium ions are less inside. The potassium ions are more inside, and the potassium ions are less inside. But the amount of positive charges due to the high amount of the sodium ions outside here will make the outside more positive than that which is inside here. Therefore, resulting into the gradient, therefore resulting into the charge of the resting potential to be negative 70 MV. And that is what resting potential talks about. When we come to depolarization, in depolarization, when the resting potential achieves its negative 70 MV, the sodium ions rushes in through the sodium channels. As they rush in, they make the inside more positive than the outside. As you see, the charges, I've placed them here. Therefore, it will rush to a point that it will achieve up to positive 30 MV. After achieving 30 MV, we're going to go to repolarization stage. In repolarization, it's like sodium and potassium, they hate each other. Look at what happened here. Sodium moved out. Sodium moved out. Potassium came in, sodium moved out. Again, sodium is coming. Potassium saying, no, bro, I'm going out. So in repolarization, potassium moves outside, leaving sodium alone. So it rushes outside after depolarization achieving positive 30 MV. It rushes out to a point that it decreases slightly lesser than the resting membrane stage, that is up to negative 80 MV. If you like the video, thank you. Comment, alhamdulillah.